Hey, welcome back to another podcast episode of the Theoretical Biophysicist. Today, I am very happy to be joined by Professor Zhang, a researcher at MIT, and we're going to be discussing one of his recent works in the Journal of Chemical Physics. It's titled Multiscale Modeling of Genome Organization with Maximum Entropy Optimization. So Dr. Zhang, thanks for being on the show.
Well, thank you for having me.
Yeah, so I guess we can just kind of dive right on in then. So, this paper is about packaging genomes. Can you kind of help frame the problem for us and maybe why you got interested in it in the first place?
Right. I guess if you think about it, the genome is actually a really kind of a very interesting and a challenging problem. Well, I guess first of all, it's biologically it's sort of has a lot of it's important because it contains all the DNA. The DNA is supposed to instruct the development and the operation of an entire organism. And I guess sort of a lot of effort has been really try to basically devoted to really understand the DNA, to understand how the information transfer occurs. How do you go from base pair to a walking and talking person? So a lot of progress has been made in that regard, and one of the sort of finding, actually that's kind of is only about maybe like a decade old, is that the genome structure is actually sort of very important for gene transcription. And so in some way, you essentially should think of a genome instead of a very long DNA molecule. It should actually be more like an enzyme. It's just like a protein. So that it needs to fold in 3D to form active sites to catalyze chemical reaction. In this case, it's basically the transcription.
Yeah, just kind of like when they did the Human Genome Project, they thought once we knew what every base pair was, they're like, all right, we figured out all of biology. But it turned out not to be the case, right?
Right. I mean, it turned out it's more complicated, and then sort of there's a. I mean, I guess complicated is sort of is also interesting in some sense. The reason I get really interested in this problem, partly because of biology, but also there's really a lot of very fascinating sort of physics and chemistry in it as well. Just to give you example, like for human, right? Our genome has over basically 6 billion base pairs. So that's about 2 meters, once you sort of fully sort of stretch it and sort of align it. And but now this 2 meter long molecule or multiple molecule is confined into a nucleus. The nucleus only has about a diameter of 10 micron. So there's a 5 order of magnitude difference in there. And so basically, so you really need to package the DNA to sort of to fit inside of the nucleus. That's kind of largely this genome folding problem. How does the cell solve this problem, and while still make sure that it's a function, right? You can't just randomly stuff the DNA into the nucleus. That's actually part of
I mean, it kind of raises the question, right? When you're packing for a trip, you just take everything that you have and jam it in the suitcase. So exactly. I guess for DNA, it's important, where things go in the suitcase.
Exactly. So it actually turned out there's kind of very interesting argument that just like we are all familiar with the Christmas lights, right? So when you just try to take the Christmas lights from the tree, and if you just randomly package it, you would easily you often find that they actually form knots, get entangled with each other. The next year, it's actually really hard to sort of set it up again.
Oh, trust me, I have a lot of experience.
Exactly. And that's actually well known in terms of polymer physics, and the reason it like to tangle up is because when polymer under confinement, those tangled or knotted configuration are actually sort of have a higher configurational entropy. But you can
so this, I guess, explicitly is kind of referring to like random tangles, because DNA can kind of like wrap itself into a knot and like writhe and twist, right? But that's not what we're discussing here.
Right. Well, I guess it is that. I mean, but I guess the idea that if the DNA formed a lot of knots, that's gonna create a lot of problem for biology. it will be very hard for for you to separate the DNA, for example, for cell division. It will also be really hard, for transcription, right? If you have a knot, then how do you unwind the DNA and sort of really then transcribe the DNA into RNA stuff?
Yeah,
So,
because I guess back to the suitcase analogy, we're not just, cramming DNA in. We need to be able to go in and pull out, each day a different pair of socks or something like that. It needs to be accessible.
Right, exactly. So it's not a completely disorder. There had to be some order in that. So basically, how does sort of biology solve this problem? That's kind of the big question. We're trying to figure out and understand here.
Okay, so yeah, it's important that we understand these genomes. What makes them hard to really study? Like, why is this such a difficult and fascinating problem?
Right. Well, it is hard to study. I guess just first of all, it's really large, as we said, right? It's it's a 6 billion base pair. That's a lot of atoms, And again, the second challenge is again, it's really really large, right? It's 2 meters. That's like a really microscopic scale. As a computational chemist, we're very good at modeling, like angstrom or nanometer scale. Maybe nowadays we can do like millions of atoms, right? That's kind of almost the state of the art. We are now very good at this studying those systems with the molecular dynamics simulation and nerves, and we have good force field. But that's just teeny tiny compared to the genome, right? So the genome has 6 billion bits. So then how do you if you want to understand the organization of the genome, how are you going to sort of even begin to think about modeling that, right? Sort of, I guess coarse graining is one idea that's very popular when people begin to sort of study larger scale problems. But then with coarse graining, the challenge here is really how do you kind of parameterize the force field? How do you really build a model that is biologically relevant? You can always simulate something, but then how do you make sure the structure you have is really actually is meaningful for biology?
Yeah, so you need like some experiment, like it's always a feedback loop, right? Experiment helps inform theory, theory helps you tell which experiments you need to do. Right. So I guess when you study the genome, what do you even try and look at experimentally? Because like we said, this thing is huge. It's not like you can just do a very easy experiment to see it.
Right. Yeah, exactly right. So I guess that's actually kind of one very exciting aspect of genome folding nowadays, because there has been sort of a lot of almost like a breakthrough in terms of experiment, where now they are actually, both in terms of high throughput sequencing and microscope images. So nowadays they can in terms of sequencing, they can basically study the whole genome. They can sort of look at the contact and to basically figure out the frequency of a different part of the genome that come in contact inside the nucleus. They call this technique called chromosome conformation capture. In some sense, it's actually similar to NMR for protein. I like to make the analogy because the NMR sort of tells you the distance between different residues in your protein, and here it's basically the same idea. There's sort of the chromosome conformation capture or Hi-C that will tell you, if you have two parts of the DNA, how frequently they will be in contact in the nucleus. So that experiment actually provides, you can sort of see, provides a lot of information about the 3D organization.
Yeah, it gives you some kind of intuition about where things lie in the system.
Right.
And so from there, you can try and build different models to at least see if you can recapture that same data.
That's right. Yeah.
So, kind of one of the main ideas of the paper is kind of this maximum entropy model, where you take some observable that you have and you build a model to kind of describe everything around it. But before we kind of get into exactly what that entails, how do you like to think about entropy?
How do I like to think about entropy? I guess that's a good question. I kind of like entropy. Because that's I feel like that sort of makes everything interesting. That's basically that's the whole idea of statistical mechanics, right? You sort of have to look at all the different configurations yeah, I mean, I would say, I mean, that's a good question. I guess I don't have a a very good answer. I mean,
well, I mean, I think the results show that people that think about this too much, like Boltzmann, usually don't wind up turning out so great. So maybe it's for the best.
Yeah.
But anyway, so kind of the idea of this maximum entropy model, can you kind of describe what exactly you're trying to do with this to kind of retie that back to experimental observables?
Right. So I guess this maximum entropy sort of, I mean, I guess this idea comes from maybe the information theory, where I think basically, the idea that if you sort of define the entropy in terms of like a p log p, and you then basically starting from there, you can sort of really derive thermodynamics. You can derive Boltzmann distribution, right? So the idea that that's kind of a starting point, and then sort of there has been a lot of work sort of using that idea to basically to couple with the molecular simulation and incorporate experimental measurements into the model. I mean, the idea that no matter how hard we try, I mean, the model is always going to be a model. There's always going to be approximation. The model never going to be perfect, right? So then the idea that if you sort of find some maybe results or from your simulation that does not agree with the experiment, you could potentially then incorporate the additional information of the experiment into your model to correct it. Okay, but then there are many ways you can think about it, sort of correcting a model to agree with the experiment. And the idea of the maximum entropy is that it basically is supposed to sort of essentially figure out a way that sort of kind of introduce the least perturbation to your model. So you basically want to kind of preserve the entropy of your system as much as possible, because when you sort of introduce additional constraints, when you introduce additional information, you're always going to lose entropy, right? You're constraining your system, or you're always going to sort of reduce the entropy of the system. And the idea of the maximum entropy is that you want to sort of bias your model in a way that that least affects the model, so that you don't want to introduce the information that's not supported by experiment. That's kind of the idea.
Yeah, I mean, we know from the second law of thermodynamics, right, that the entropy is always kind of increasing. So we want to find whatever probability distribution that we have that's going to maximize their entropy.
Yeah, exactly. Yeah. I mean, I guess so. I mean, this is also like similar to what people do, like in terms of maybe machine learning, where where it's basically minimizing the so-called Kullback library divergence. It's really the same idea.
Yeah, and the Kullback-Leibler divergence is the difference in information between two systems. So
exactly,
yeah. So I guess we have this methodology now for maximum entropy, where we can try and rebuild out probability distributions. So I guess the hope then is to basically take these distributions or create these distributions and try and recreate the high C data that we have.
Right. So I guess I mean, if you recreate well, I guess what you want is you create a distribution that matches with the high C, right? But this distribution now is a conformational distribution. It's basically an ensemble of structure for your genome. So the idea is that you are basically now, but now what you see that you have actually much more information than high C, right? High C is basically a two-dimensional contact map in some sense. So it's a 2D information. It does not, but the idea that so what's nice about the maximum entropy is that you can actually get out more than what you have in the experiment in the high C, because the idea that you can also in addition to the high C, in addition to the experiment, you can also basically provide more sort of known knowledge or aspect or information, if you like, into the model. For example, we know the genome it's made of polymer, right? So that they're sort of, they're connected chains. I mean, this by just being a polymer itself, they're already sort of, put a lot of restriction onto the structure, right? So the idea that then the distribution we sort of we corrected with the high C, now actually has more information than the high C because we incorporate all the polymer nature of the genome. So now we can actually look at the 3D structure. Yeah.
That's the hope with the model, right? Is you want to be able to get something out of it. If I could only give you high C data, you would go, I already have high C data. I don't need high C data.
Exactly. Right.
So I guess, like what specifically are you trying to tune then with this model? We talked about it being a polymer. So, people have looked at protein folding and polymer folding for a while. You just try and use a very similar like model.
Right. So it's here, it's basically a coarse grain model for the genome. As we said, the genome has 6 billion base pairs. We cannot afford to simulate that, so we basically have to use a coarse grain model. They kind of bead on the string model, and each bead it's typically represent thousands or maybe sometimes even million base pairs to make the model tractable. So then what we really kind of parameterize, using this maximum entropy algorithm from high C, is that you can think of it as basically a force field for the genome. It just sort of tells you, because the high C give you the pairwise frequency or the frequency per pair of DNA to be in contact, what we can do is basically from that we can infer or derive the interaction energy, the energy between pairs of DNA. So you can basically sort of derive a potential energy function for the genome or the force field for the genome.
Yeah, because with that energy, we know that pairs that have a very high repulsive energy are not going to be close to one another, and you can see that in both high C and the model.
Exactly. Yeah. If you see sort of two pairs that have very high contact frequency, then presumably they would have high probability as well.
So what's kind of the when do you know that you've gone far enough with this model that you can say, okay, we've gotten something like meaningful out of this? Like what's kind of the big pie in the sky you're after?
What's the big pie in the sky? Right. I guess I mean well, I mean, I guess our in the end our dream would be to really try to see whether we can actually predict the genome structure, instead of always try to learn the structure from experiment. Because if we can really predict the structure, that would sort of I mean, I guess along the way we're trying to basically learn the mechanism for what kind of dictates the genome structure, because there's still a lot of unknown regarding sort of how the genome is really set up and everything. Like we said, it's not random. So there's a lot of molecular factors that actually play the role. But then the end goal is really sort of if we understand all the mechanisms, then can we predict the structure for the genome? Just like now that you can more or less predict protein structure from the amino acid sequence, could we sort of do a similar thing? Could we predict the genome structure, from the underlying DNA? Maybe there also additional, they call this the epigenetic information. Could we also, make those predictions?
Yeah, and I mean that epigenetic information is super important, like on an individual basis, right? Like you can stack the deck however you want, but you still might kind of luck of the draw, get an ace every now and then.
Right.
I guess it seemed like in the paper you guys talked about, I think what's kind of an interesting behavior. It's kind of this idea of phase separation in the genome, which is you wouldn't really expect. I have only about 4 base pairs. Like, why is this phase separation occur? Like, is there any rationale for it?
Right. Well, I mean that's a great question. I mean, I guess I would say the answer is probably not settled at this point. I mean, there are actually many ways for you to actually have phase separation. Well, it turns out that sort of from polymer physics, the long polymer is actually really hard for them to mix, to mingle. For small molecules, they're actually very easy to mix with each other. But for polymers, it's much harder for them to mix, and it's because the mixing entropy actually has a prefactor. If you look at some of the textbooks, the mixing entropy for polymers is scaled by 1/n, where n is the length of the polymer, meaning that the driving force, which is sort of for the molecule to mix, is actually very small for polymer. It's very substantial for small molecule, but they actually sort of very small for polymers. So it's actually really hard for polymer to mix with each other. So that's why that kind of the first layer, phase separation, is that all the chromosomes, they actually tend to they form the so-called territory. They don't really mix with each other, like so I guess I like to joke that instead of thinking of this as a bowl of spaghetti, you should think of it a bowl of meatballs. They're kind of separated from each other. That's chromosomes.
I'm sure there's a lot of Italians out there like, oh, no.
Yeah, but then on top of that, like you said, there's also phase separation because we know that there are epigenetic modifications. These epigenetic modifications that basically sort of essentially modify the chemistry of the DNA now. Different part of the chromosome, they have sort of very different modifications, and those modifications that they can have actually very different interaction. They typically have it's more like a block of polymer. Now the similar modification have attraction with each other, but they maybe have like a repulsion between different modifications.
Yeah, I think that idea of a block copolymer is kind of interesting. And in the paper you guys had shown where, we need to have, maybe more than just an AB, there might need to be an ABC block copolymer. And, it's kind of the question of how many blocks do I need to have before, I run out of stuff.
Right. That is a great question. I mean, that's actually the thing that we are trying to figure out. I mean, that's a part of that. I guess that's a part of where it's actually very important to compare with the experiment. Because I mean, you sort of that's actually kind of what's nice about actually our method, because by doing this sort of maximum entropy algorithm, you can essentially infer all the parameters from PICE data. So basically, there's no parameter tuning in some sense. Because all the parameters are kind of fixed, but based on experiment data. So then the idea is that you can essentially use this as a way to screen hypothesis, right? If you like you said, suppose I just come up with a very simple model for the genome. I think that's the AB block of polymer with two types. And then I can basically figure out all the interaction in the model from high-C data. But then the question is that now I have a model for the genome. How much does it really explain about the known sort of aspects of genome organization from previous experiment, right? There's sort of a image experiment which sort of tells you how the genome really organizes. So, so you can sort of basically kind of cross-validate your model with the other experimental data. That's not provided for parameterization. So then you can.
So do you think it might be possible, kind of with protein folding, Ken Dill came up with this idea that, proteins fold because we have hydrophobic and hydrophilic. Do you think we'll be able to ever collapse something into, kind of that great of a simplification?
I mean, there is a hope. I mean, that is a good question. I mean, I guess, this idea. I mean, part of the challenge it's very complex. Sort of, because in addition to this, maybe in the block copolymer picture where you basically have just a passive interaction, the equilibrium model. There's also non-equilibrium sort of activity that's going on in the system, and the non-equilibrium motor themselves could also drive the phase separation. So but I mean, it's possible that multiple factors that contribute together to this final phase separation. But I think now that we actually know a lot more about maybe the chemistry, underlying chemistry that could drive the phase separation, that could give you this sort of block copolymer. I guess the simplest picture is like heterochromatin, euchromatin. And, there is some truth to that. That could phase separate, but they may not be the only sort of ingredient for this phase separation.
Yeah, I think that's kind of a good segue then into the next part. Chromosomes, they aren't just it's not just like DNA is the only thing in the nucleus. Like there's, we have all these other different chaperone proteins and things like that. So, when we go from this huge scale, where we're modeling, maybe 50,000 base pairs as a single bead into something, where we now have the genome that's wrapped around, the different histone proteins that form the nucleosomes. Like what challenges do we have there that we didn't have on this larger scale now?
Right. I guess I mean I guess right. I mean we are also very interested in sort of looking at the chromosome at a very sort of fine scale, at almost we call it atomistic or near atomistic level, because there we really can sort of ask a lot of questions that you were saying. Look at the mechanism, right? Sort of what are the sort of physical chemical interaction that really sort of drive the phase separation, etc. So I mean in principle there we're actually using more or less sort of standard approach that people use nowadays. We basically use the molecular dynamic simulation with a sort of corresponding force fields, and there's a lot of force field has been developed in that regard. The main challenge that we face there is that there's a lot of disorder protein that actually sort of that contribute to phase separation. And the disorder protein, they're actually quite tricky to model, and the reason is that I mean the disorder protein only sort of kind of get a lot of attention very recently, and a lot of the force fields that were developed previously, they were kind of mostly targeted for protein folding. Right? They want sort of predict protein structure. They want to collapse the protein to sort of look at the tertiary structure. But disorder protein don't really have those tertiary structures. So then one of the challenges is that you cannot really just directly apply existing force fields to look at disorder protein, because they typically tend to over collapse those proteins. So we sort of have to kind of figure out new ways to parameterize the force field, and then also important to make sure it works for both order and disorder proteins.
So yeah, I guess the dream there is just be given the primary sequence for protein and just predict the shape, no matter whether it's ordered or disordered.
Right. That is kind of the dream. I mean, I think basically I would say there has been a lot of a lot of smart people have thought about this problem, right? They have thought about the parameterize the force field for proteins. But I guess I would say the only sort of caveat is that those I mean I think in principle those algorithms could be well used to parameterize force field for disorder protein. Just they hadn't been really sort of tested or validated or benchmarked on disorder protein. So we're basically sort of saying that we're sort of now can we have engineer, or can we develop an algorithm that can allow you to parameterize order and disorder proteins.
So that's when we, reach back in our toolkit and pull out this maximum entropy idea again, and we retune our parameters now based on some experimental observable.
Right. Yeah. That's actually a good point. I forgot to mention. Yeah. Thank you for asking that. So it turns out that it's actually a little bit tricky to parameterize the disorder proteins, or at least to parameterize systematically. And the reason is that for folded protein, there's the energy landscape theory, which I really like because that was kind of introduced by my postdoc advisor. So
that always helps, yeah.
Right. So the idea that the reason protein can fold reliably into this sort of tertiary structure is because there's a so-called energy gap. So the energy of the sort of folded native structure, it's much lower compared to this kind of unfolded disorder structures, right? So then that idea that you have this energy gap actually can kind of allow you to can be turned into a algorithm, which then allow you to sort of parameterize the protein force field. I guess we're all talking about a corresponding force field, because all atom force field is kind of a different story here. So this, but then the problem is that that energy gap idea does not really apply to disordered protein, because by definition they don't really fold to a unique tertiary structure. So you cannot use that idea to parameterize the disordered protein. And I would say sort of there really hasn't been a sort of a very good way to systematically parameterize cross-content force field in kind of almost before our study. But what's nice about the maximum entropy is that now it actually with additional tweak, we basically have to sort of once we have to sort of couple this maximum entropy with a reparameterization step. But what that does is that basically that allow you to sort of systematically parameterize the force field now, to make sure that actually agree with experimental data, and you can also sort of essentially couple this maximum entropy algorithm with all the previous algorithm for folded protein. You can easily couple them together to sort of to make sure that now the force field can work for both ordered and disordered proteins.
So yeah, that's interesting. We're using kind of this entropy to help bridge the gap then between disordered and ordered proteins.
Right.
So I guess it kind of leads nicely, and I guess kind of the last bit of the paper you're talking about kinetics in some sense. So you have this genome, it's not just a static structure. So when you do maximum entropy, you're typically talking more about like an equilibrium distribution. So how can we take this idea now and apply it to something that's dynamic?
Right. I mean, the dynamics is always tricky. I guess the maximum entropy it really does not sort of actually incorporate or does not really concern that much about the dynamics. But there has been sort of a generalization that was introduced by Ken Deal called maximum caliber. It basically sort of kind of in a sort of similar idea, just like the maximum entropy sort of takes the ensemble averages from experiment and sort of incorporate that into a model to sort of improve statistics. Maximum caliber allow you to sort of take dynamic information and sort of incorporate into your model and potentially help you to improve the kinetics of your model as well. I mean, the kinetics of the genome is another really fascinating sort of area. There's a lot of questions.
Yeah, I mean, kinetics in general are really difficult. So
that's right.
I guess
that's why we haven't done much yet.
Still a lot of work for the future people out there.
Exactly. Yeah.
So I guess what's kind of interesting about it is with maximum entropy we have like this nice high C map. We have like reasonable experimental data that we can compare to. So with maximum caliber, then what exactly are we trying to compare to experimentally to refine this?
Right. I mean, so there are that's sort of we basically come to now that the advancement of kind of almost live cell imaging. So now with live cell imaging, they can actually essentially track the dynamics of the different part of the genome and sort of basically monitor the the movement of the chromosome inside of the nucleus. And there has been a lot of studies that sort of show that the chromosome has this kind of subdiffusive behavior, and recently there actually has this very sort of nice paper from I think from the Little Whisker, I think, yeah, from her lab at NYU. They showed actually very interesting that the genome kind of has a crossover between sort of like a fluid state or liquid state to this gel-like state. There's a kind of a lot of very sort of interesting flow dynamics going on. So those kind of information would be useful to maybe as a way to both benchmark your model, sort of see whether you are getting meaningful kinetics, or sort of maybe to figure out somehow maybe to incorporate that information into the model. So
okay, neat. So I guess, is there anything else that you kind of wanted to highlight or discuss about?
I think we covered pretty much, but I mean, I just wanted to say, I would say, the genome folding is a really an interesting problem, and there's a lot of open questions. I would say, unlike protein folding, which has been studied, for many years, there's a lot of open questions. There's a lot of opportunity for discovery, I would say.
Open questions are good. That means there's a lot of grant money, hopefully, out there.
Exactly, yeah. And so that's why I think I would welcome sort of more people to join in the field.
So if you did have someone that wanted to get into this, what would you kind of point them to kind of maybe get their feet wet and get a better feel for the magnitude of the problem? Other than reading the paper, of course.
So, I mean, I think that there's a lot of discussion sort of on phase separation nowadays. So I mean that's why I think like people with a physical chemistry background really could actually help understanding or improve understanding of the genome, because it turns out that that's kind of almost like a revolution in cell biology, which they found that a lot of sort of the stuff in the cell are kind of like just like liquid droplets. And there's a lot of open questions regarding how they form, and sort of what sort of control their number, what control their specificity, composition. There's a so it's kind of really sort of exploding idea, exploding field, and there's also liquid droplets sort of that actually played a lot of role in both the genome organization, but also in maybe in genome function, genome transcription. So there's so I think there's a lot of open question in that area. I mean, there's a kinetics, like we said, there's a gelatin, another sort of really fascinating area. So I think sort of people with sort of chemistry background really could actually make a impact here. I mean, I would say don't get sort of maybe overwhelmed with the biology initially. I mean, it's kind of you might think that it's a genome, sort of it's also so complicated, a lot of stuff there. But there's also a lot of very sort of well-defined and interesting physical chemistry problem in this area.
Yeah, I mean, you can definitely get bogged down in all the terminology, the different cell phases, the difference between chromatin and chromatid. Yeah. I think I still have to look that up every time.
Right.
So I guess, kind of what keeps you curious and keeps you going then?
What keeps me curious, keeps me going? Well, I guess there's just so much unknown out there. I mean, like I'm sort of like I think today I was sort of reading up this paper, which is I think they were studying basically the colloid sort of phase separation, which is actually it's fascinating, and because colloid phase separation can sort of easily lead to gel and glass. And it turned out that actually had a lot of analogy to what we are looking at in terms of the genome as well. So there are just so many interesting questions that's out there. So I think that sort of
there's still a lot of space left on your head you can scratch, right? You're not
exactly. Yeah, I don't know this. I don't know that. I mean, that's just because the genome, it's a multi-scale. It's really a truly multi-scale problem. Sort of both in terms of time scale, in terms of length scale. So there's.
I mean, yeah. At the beginning, we said it stretches, 2 meters, and then it's right. We're looking at a single nucleus, which is 10 microns, right? So there's a lot of scales there.
Right. And also, I'm not tender yet, so that's also keep me going.
Yeah, that'll definitely keep you at the coffee pot on those long days.
Exactly. Yeah.
So yeah, I don't have anything else. Thanks so much for being on the show, Dr. Zhang.
Well, thank you for having me. Yeah, it was a lot of fun. Yeah.